All right, so today we're talking about chapter 9, which is sexual reproduction and meiosis. Remember, we've already talked about mitosis, and there's some differences between mitosis and meiosis. So let's just roll these out one more time. Remember, meiosis is going to have two cell divisions. You're going to have meiosis 1, which is going to separate the homologous chromosomes from one another. And then in meiosis 2, you're going to end up separating sister chromatids from one another. And so I know this doesn't make sense now, but it will at the end of lecture today. Also in meiosis, what we're going to do is we're going to start with a diploid cell. And that diploid cell is going to undergo meiosis 1, the first round of cell division. And then that's going to create a haploid cell, or a cell that has half as much genetic material in it. And then it's going to go through another cell division, and that's going to make 2 of those. And then it's going to undergo meiosis 2 in order to produce 4 of these haploid or 1N daughter cells. And again, these are all going to be genetically diverse. Unlike mitosis, which you're just making a clone of a cell. But in meiosis, you're making genetically unique gametes, either your sperms or your eggs. Yes?
Can you go do that again?
Yeah. I mean, I'm just kind of giving a brief introduction. That was my introduction. So.
It was a fast introduction.
Well, I'm going to talk about it again. Trust me, you'll be good. All right, so when we're talking about our DNA is linear, right? So we've got lots and lots and lots of DNA. So there's our DNA. And along those DNA, you have individual genes. You know, genes for this, genes for that, various different, genes that give rise to various different types of proteins. And so and by the way, these genes are arranged on chromosomes. So an adult human, we have 2 sets of genetic information, right? We have a set from mom and we have a set from dad. So that means that we are diploid. So a diploid organism is going to have 2 sets of genetic information. We've gone over this before, right? Diploid and haploid?
Mm-hmm.
Pretty sure we have. Okay. Now our chromosomes, like I said, we have 46 chromosomes. So what would a haploid version of our genome, how many chromosomes would it have?
23.
It'd have half as much, right? So haploid would be 23 chromosomes. And so of the set of 23 chromosomes that we inherit from mom and dad, 22 of them are going to be normal chromosomes. We call those autosomes. Whereas the remaining pair, these are all pairs of chromosomes, by the way. And so in addition to the 22 autosomes, so these are chromosomes 1, 2, 3, 4, all the way up to 22. The other pair is going to be your sex chromosomes. Either your XX if you're a female, or your XY if you're a male. And so 1 pair of sex chromosomes plus 22 pairs of autosomes equals our genome, our 46 chromosomes. And so in those 46 chromosomes, remember we're starting with DNA from mom and dad. So that means we've got 23 chromosomes from mom and we've got 23 chromosomes from dad. Well, homologous chromosomes are going to be the same chromosomes but from different sources. For example, like chromosome 2 from mom is homologous to chromosome 2 from dad. It's because they're the same chromosome. If they're the same chromosome, that means they have the same genes running along the length of it. They just might have different versions of those genes, but it's still chromosome 1 of mom compared to chromosome 1 of dad. So they're going to be very, very, very similar, again, because it's the same genes. It's going to be tweaked a little bit depending on what versions of genes your parents have for a particular trait. And so these homologous chromosomes, these are going to come into play in the first round of meiosis. So I'll be coloring in a lot of chromosomes today. And so when you see something like this, that's a homologous pair of chromosomes. All right, they have the same gene here versus here. They got the same gene here and here. Same thing here. So for instance, this might be the gene for eye color. Your mom might have blue eyes, and your dad might have brown eyes. So it's the same chromosome, it's just different versions of the gene. So you can either have the brown-eyed version of the gene or the blue-eyed version of the gene, but it's still the gene for eye color, which is why these are going to be homologous. Homo means the same, and so if we say homologous chromosomes or homologs, that means that they're the set that came from mom and dad. And by the way, I keep talking about these different versions of genes. Those different versions are called alleles. So you can have an allele for blue eyes, you can have alleles for brown eyes, you can have alleles for green eyes. All alleles they're just different versions of a particular gene. Just like I could have an allele for brown hair or red hair or blonde hair or any other sort of trait that acts in this type of fashion.
Just how that gene is expressed, right?
Yeah, it's how that gene is expressed, exactly. Yeah, Nancy?
So with the gene number, and everyone's gene 1 is the same thing, like
Yeah.
for example is I. So okay.
Yeah, all the chromosomes are the same. I mean, when we sequence the human genome, everybody's genome is going to be a little bit different, but they averaged a whole bunch to get kind of, at least a baseline. Now, like I said, everyone's is going to be a little bit individual, but for the most part, humans share probably 99.99999% the same sequence. It's just a nucleotide here, or maybe a couple nucleotides here, a few over there, but overall very small amount of the genome. So that's why we call them homologous, because they're the same chromosome.
So if we're all pretty much similar and the same, how would you determine your DNA from my DNA?
Just by sequencing it. There's all sorts of different markers and, little signs in the DNA that determine, oh, this is this in you, but this is this in me. You can actually compare sequences with one another. You can compare human sequences with one another, but most of them, like I said, are going to be pretty similar. So if we wanted to compare our DNA, all we'd have to do is go get a DNA sequencer, run it a couple hours later, print it out, and we'd know our sequence of DNA. Which, by the way, I don't know if y'all know this or not, and I don't know if you're interested, but there's a company out there that's called 23andMe. It sequences your genome and looks for health risks for like $100.
23andMe?
23andMe. And, I definitely don't work for them or anything. I'm going to do it because I think it's cool. Because it'll tell you, like, if you have markers for certain diseases in the long run, it kind of gives you, like, a heads up. Like, it explains the blueprint of your body, maybe what you should be expecting for versus what you may not have a risk for. So I'm just throwing that out there. No extra credit if you do it. All right, anyways. Okay, so there's a way that we can look at all the different types of chromosomes in the humans, or for any organism for that matter. And the technology that's used to do this is called a karyotype. And all they do is they take all the chromosomes and they stain them with special dyes so that certain regions of certain chromosomes are stained this color and other regions are stained another color. So you get these kind of multicolored chromosomes. But because there are certain dyes that stain certain regions, you can match up, oh, this has to be 2 Ys because that both is pink and that both is there and that's all the same and that's all the same. Plus, let me get there. Plus, every chromosome is a different length. All right? They start off being really, really, really big. So chromosome 1 is the biggest chromosome. And then chromosome 22, this is the smallest chromosome. So they start off from the largest size and get smaller and smaller and smaller. So you can pair them up based on size and then also on the dye. And then they'll also, of course, show your sex chromosomes as well. But this karyotype, this is used for looking for kind of like gross abnormalities in chromosomes, like Down syndrome, for instance, is you have an extra copy of chromosome 21. So that would be really easy to spot on a karyotype if you've got a whole extra chromosome. You've got 3 chromosome 21s instead of only 2. So these karyotypes are ways that we can match up and look at kind of these big changes that may or may not happen in chromosomes. Yeah, Daniel?
Is that a drone?
That's a girl, because she's XX. Girls have 2 X chromosomes, and a guy has an XY. And by the way, just to let y'all know, the default for human development is female. If you have 1 X chromosome, it's still going to be a female. It's the Y that determines whether or not it's going to be a boy. But the Y chromosome is one of the smallest little chromosomes because there's very little genes that make a male a male. That's what I'm saying. The body is female by default, and only by those few, I don't know how many genes, but it's not very many, testosterone being one of them, and the sex-determining gene. But anyways, that's why the Y chromosome is so small, because y'all don't really contribute a whole lot to it.
All right.
Yeah.
What about the hermaphrodites?
There's lots of different ways to get that. That's usually a developmental thing.
Oh,
And there's all sorts of other, like you can have diseases that have 3 X's or 2 X's and a Y, or Y and 2 X's, or any number of combinations, XXX, YYY, that have all, that are some are normal, some are affected others, and so that kind of falls into that group. Yeah.
Is there a reason for their structure the way it is? Like some are perfectly straight, but then
No, all they did was literally like use a teeny tiny little thing and move around a piece of DNA. So it's nothing significant about, it's just thrown up there. That's all it is. Okay, so we've already talked about sister chromatids, right? Sister chromatids are genetic copies of one another. So this is the exact same copy as this. Now homologous chromosomes, they're going to be the same chromosome. So say chromosome 1 from mom and chromosome 1 from dad, those are going to be a pair. And so this is going to have its own sister chromatids too. So these 2 are going to be identical to one another, and these 2 are going to be identical to one another. Now they're both chromosomes 1, but because they came from 2 different sources, they're not going to be genetically identical. So they're homologous chromosomes, which have the same genes in the same order, but are going to have different versions of those genes, or different alleles of those genes. So in this example, let's just make up a trait for this. In this particular organism, let's just say that big B represents brown hair. So this individual, and I know y'all don't understand the A's and the B's yet, but it just represents a gene, that's all it is. Anyways, this individual represents brown hair. And this individual would represent blonde hair. So it's the same, they're both homologous chromosomes, but they have they're not genetically unique. Because they're going to be a little bit different. Because mom might have, brown hair, and dad might have blonde hair. Still the same gene, just different versions of the genes. So sister chromatids are exactly the same. So these 2 are identical, and these 2 are identical, but this one and this one is not identical, because it's from a different source. And so when we're looking at meiosis, it's going to be real similar to mitosis. The only difference is we're going to add in an extra cell division. So in mitosis, remember we replicated our DNA and all we did was separate the sister chromatids, right? Well, meiosis 2 is going to look just like mitosis. All we're doing is separating the sister chromatids, just like we do in mitosis. Now the thing that's going to be different is in meiosis 1, we're going to separate the homologous chromosomes from one another. So in other words, we're going to take mom's chromosome to one side and dad's chromosome to the other. And then eventually those will end up in these new cells. So at that point, if you take chromosome 1 from mom and chromosome 1 from dad and put them in different cells, so here's mom's, here's dad's, these are going to be haploid now, right? Because diploid means that you have both versions of the chromosome, one from mom and one from dad. But if you only have one or the other, then you're going to be haploid. And we'll talk about this a little bit more. I might be getting a little ahead of myself, but just want to kind of give y'all a heads up. All right, so when we're talking about meiosis, we're talking about the sex cells, the sperm and the egg. That's the whole purpose of meiosis, is to generate the gametes. And the gametes are always going to be haploid, which makes sense because if you have an egg that's haploid, and then you got your little spermy sperm that's haploid, and then once fertilization occurs, this whole organism is going to become Diploid. Yeah, but I mean, it will become solid. They'll merge together. I mean, that's what happens once this penetrates. He'll make his way in, and their DNA will combine and form a diploid developing organism, which in this case is called a zygote. So a zygote is what you get immediately after fertilization. It's that first cell that's going to be diploid as a result of two haploid gametes coming together. And so, and again, when comparing it to mitosis, remember mitosis, we're just making genetic copies of a body cell. Whereas in meiosis, we're making genetically unique gametes in our sex cells, or forming our sex cells. Okay, so life often alternates between meiosis and mitosis. This is true not only for humans, but for other organisms as well. And so this kind of goes into the different life cycles that are observed among organisms. So when we first started out, we were just one little cell. Right after fertilization, we were diploid. And then we started making more copies of ourselves. How do we think we got from one little cell to what we are now? Through lots and lots and lots and lots of mitosis. So through development and growth, we undergo mitosis until we end up being adult, being what we are today. And then when you get into puberty, that time, then you're going to start undergoing meiosis. So you're going to start making your sperm, making your eggs. And so those eggs and that sperm, those are going to be haploid again. And so then when you find somebody that you love very much and you have sex and fertilization occurs, it starts back over at the zygote. So it's this continual cycle of life. I mean, all of our cells in us right now, minus the egg and the sperm, are going to be diploid. So our body is always undergoing mitosis, always, ever since we were born. Meiosis, that's only going to come on at certain times due to certain hormones. I mean, males produce sperm in their entire life, whereas females, they make all their eggs early, actually in the womb, and they don't start releasing until, 12, 13, 11, depending, 8 in some cases. But there's the take-home message is that there's this alternation between meiosis and mitosis in a living individual. And so we already mentioned that meiosis is going to have two different cell divisions. The first one's called meiosis 1, second one's called meiosis 2. Yes, it goes in order. Sounds kind of logical. And so in meiosis, we're going to start with one cell that's going to be diploid. It's going to have both sets of both parents' chromosomes. And then we're going to separate those homologous chromosomes that we talked about, separate those homologous chromosomes in meiosis 1. So after meiosis 1, we're going to end up with two cells, and they're going to have either a chromosome 1 from mom or either a chromosome 1 from dad. They're not going to have both, which is why they're haploid. And then from that point, they'll go into another round of division, meiosis 2. And again, this is going to look exactly like mitosis, because all you're doing is you're separating the sister chromatids from one another. So in meiosis 1, you separate the parental, the homologous chromosomes, and then in meiosis 2, you separate the sister chromatids. And so after meiosis 2, you end up with 4 cells, each one of them genetically unique, and each one only containing one set of DNA. And so there's going to be 2 cell divisions in meiosis, but of course there's only 1 cell division in mitosis. And so here's again kind of the layout of what we're looking at during meiosis. So these in blue, these represent, all the different autosomes, and I guess this is representing the sex chromosomes right here. But anyways, so you have your diploid human cell. All right, it's diploid because it's got matching chromosomes from mom and dad together. And then in meiosis 1, this is separating the homologous chromosome. So if this is chromosome 2, and this is chromosome 2, that means there's not both chromosome 2s in one cell, which is why it's haploid. And same thing for this blue guy right here. His homolog, or his buddy, is in a totally different cell, which is why these are haploid and only have one set of genetic information. And then in meiosis 2, after we separate the homologous chromosomes, then we're going to separate the sister chromatids from one another. And so you're going to remain haploid. You're just going to have half as much DNA as you started with before meiosis 2.
Picture's helpful.
The picture is helpful? Yeah, no, it does help. So in meiosis 1, you're separating the homologous chromosomes. And then in meiosis 2, you're separating the sister chromatids. And so just like the cell cycle for mitosis, there's also a cell cycle for meiosis. So you're still going to have a G1, an S, a G2, but instead of a mitosis followed by cytokinesis, you're going to have meiosis followed by cytokinesis. And so that M phase is actually made up of meiosis 1 and meiosis 2. And so each stage has their own prophase, metaphase, anaphase, telophase. So if you see like telophase 1, that's talking about telophase that happens in the first division of meiosis. Yeah, Dana?
Never mind, I'm sorry.
Okay, so, and a lot of these mechanisms are going to be extremely, extremely similar to what happens in mitosis. So hopefully this will be somewhat a review. But there is going to be something that's going to be totally different from mitosis, and it's something that we should address right now. So we have these homologous chromosomes, right? One from mom, one from dad. And before we separate them in the first meiosis, they're going to actually switch pieces of DNA with one another. So what's going to happen is these non-sister chromatids, right? These two are sister chromatids, and these two are sister chromatids.